Let's take a closer look at different ways in which crossing over or genetic recombination can happen. So here we have our homologous pair of chromosomes again that are crossing over, and we get something that looks like that. And this is called a single crossover, for the simple reason that they're crossing over once. But sometimes these chromosomes can get, again, kind of close together, and they might cross over in that same exact spot a second time. And then we're actually back to square one, because now the chromosomes revert to what they originally were. So it might not look like crossing over ever happened, but it actually happened twice. So this is called a two-strand double crossover. And it's called a two-strand double crossover because, well, it involves two strands. Let's actually highlight the two strands that were involved. So it was this strand and that strand, and it's called double because crossing over happened twice. Let's look at another scenario. So again, we're going to cross over our chromosomes. So we're going to see that that's the first incident of crossing over. I'm actually going to label it 1. I'll label the first one also. That was the first incident of crossing over, and then we had a second incident of crossing over. So anyway, back to our second scenario. Let's say that these two chromatids now cross over. So that can happen in a few different ways. Let's go through 2 ways in which that could happen. Maybe they cross over exactly at this spot over here, and in that case we're going to get a set of chromosomes that looks like that. Or maybe not. Maybe they're going to cross over at a spot like somewhere over there, and in that case we're going to get a set of chromosomes that looks like that. And again, that was our second crossing over. So we just described something called a 3-strand double crossover. And it's called a 3-strand double crossover because, well, in this case, 3 chromatids were involved, 3 strands. So let's just highlight them. I'm going to highlight them in the beginning pair just because it's easier. So it involved this strand and that strand and that strand. And again, a double crossover because crossing over happened twice. So hopefully you're starting to get the idea that crossing over can happen in many different ways and a number of times. Let's look at a third scenario. So again, we're going to have crossing over happening one time, and then we're going to have it happening again, but we're going to say, well, maybe crossing over now happens between these 2 chromatids and also happens between these 2 chromatids. You're going to see we're going to involve all 4 strands this time. The first scenario we'll look at is, well, maybe crossing over and both of them happens right at that spot. So in that case, we're going to get a set of chromosomes that looks like that. But maybe not. Maybe crossing over, let's say in the purple chromosome, happened over there. Then we get a chromosome that looked like that. And then maybe in the blue chromosome also, maybe crossing over happened over there. Then we'd get a set of chromosomes that looks like that. And what we just described, well, this is called a 4-strand double crossover. So in this case, we involved all 4 strands. This strand, sorry about that, this strand, and this strand, and this one, and this one. And again, it was a double crossover because crossing over happened twice. So the point of showing you all this is to help us understand that crossing over can happen in many, many different ways, in many different places, and a number of times. And remember, it's not just happening to one pair of homologous chromosomes, it's happening to all of the pairs of homologous chromosomes. So crossing over is a great way to shuffle around alleles and come up with a whole new combination that didn't actually exist before. So it's a great way to increase genetic variability. A question you may have been thinking about in our discussion of recombination is, does recombination happen between the sex chromosomes? And your intuition might tell you that that might not be the greatest thing. Let's see why. So here we have our X chromosome and our smaller Y chromosome in blue. And I'm just going to stick on a gene onto the Y chromosome. And remember, this is a Y chromosome that's duplicated because we're looking at the chromosomes as they appear in the beginning of meiosis. Anyway, this is the Sry gene. And the Sry gene is basically responsible for the initiation of male sex determination in humans. And let's say that above this point, these two chromatids swap material and recombine. So that would mean that we suddenly have on the X chromosome some of the Y chromosome, including that Sry gene. And then we're going to have some of the X chromosome over here. Let's say this happens. So this is going to be pretty problematic because the gamete that gets this Y chromosome is going to be in trouble, because when that's fertilized, it's, well, supposed to become a male because it has a Y chromosome, but it's actually missing a very important gene, the Sry gene, that's going to turn it into a male. So you can see that this can lead to a whole host of problems. So let's see what actually happens between the X and Y chromosome. We have at the tips of the X and Y chromosome, I'm going to shade it in this magenta, we have these regions called the pseudo-autosomal regions. And crossing over does happen between the pseudo-autosomal regions, so it could happen between these spots, provided that it's within the magenta shaded area. However, crossing over generally does not happen between the rest of the X and Y chromosome. So this part of the Y chromosome, that is the male specific region. So crossing over generally does not happen between the male specific region and the rest of the X chromosome. It usually only happens in the pseudo-autosomal regions.